Buenos dias. I apologize. I will give my lecture in English, and my Spanish is not very good. And I hope to be able to talk slow enough so that the interpreters can translate, I think, the lecture so that it's something that's enjoyable for everybody. So thank you so much for this invitation, to Dr. Navarro for extending the invitation, to Dr. Albaru, to Mara, and some of the very good friends that I see in the audience. I'm delighted to be here. So the topic of today's talk is an omic approach to transplant monitoring. And I'm just going to take you through a little story of the last 15 years of work in our group. That has led us to understand that if you want to monitor accurately the immunological threshold of a transplant patient such that you can customize the treatment for that patient, then you have to be able to monitor for danger as well as safety. And in a transplant recipient, that means you have to have a non-invasive means to monitor for both rejection and some form of acceptance, something that we call PROP or operational tolerance. So the response to alloantigen in all organ transplantation really requires lifelong immunosuppression because alloantigen is essentially dealt with blockade by conventional immunosuppression. But you really have the following kind of cascade of events that occurs in the immune system of a transplant recipient. Largely, the big crux is the activated T cell, but you can see that the immune response can either skew towards graft rejection and perhaps a second set of graft rejection in the people who have an immune threshold that requires more immunosuppression. Or you can have a skewing of the immune response on the other end of the spectrum to people who may approach immune deviation, perhaps the expansion of regulatory T cells, anergy, whatever the mechanism that leads to actually acceptance of the allograft. And we know that in a small subset of patients, this acceptance of the allograft can also occur with very, very low and almost complete absence of immunosuppression. Not something that a physician proposes for the patient, but if we can understand the two ends of this spectrum, the danger and the safety, perhaps we can use these markers to titrate immunosuppression prospectively in our patients. So this is, I think, a really strong clinical unmet need for all of us. So I will take you through really the research that led to our understanding that perhaps a future of having a blood test that will allow us to do this for our patients is a reality. So the first part of the story that I'm going to present is really an accurate non-invasive test that was done in blood using gene-based analysis to assess for acute rejection in a transplant patient. And this is a period of research that has actually taken 12 years, so probably the most expensive single publication, I think, that's come out of our lab, because the entire 12 years of research finally led to one paper. The target that we had set for ourselves is the positive predictive value and negative predictive value of any test that we develop has to be high, has to be at least greater than 85%. Maybe or even in the 90%, so that the physician has a lot of safety in being able to use the test to actually change treatment. So we use the approach by actually stepping back and saying that even though there are a lot of published markers in the literature that track with acute rejection, clearly we know a lot of the genes that exist in the literature are also influenced by infection, by immunosuppression, by gender, by race. And we have published data from our group, as well as other people have, that the majority of genes in peripheral blood are actually influenced by other factors than acute rejection. So one of the strongest confounders is actually recipient age. It can be the white count of the patient, the hematocrit of the patient. So to just go back to the published literature and take gene-based analyses from small single-center studies probably is the reason why we have not been able to bring this forward as a clinical test. So we stepped back, and 12 years ago we decided that we were actually going to scan all the genes in the human genome in an unbiased approach. And so PAX gene tubes were used. A blood test was taken at the time of an allograft biopsy. All the biopsies in the study that I'm going to show you here were read by a single centralized pathologist to obviate the interpathology read variability, and the samples were processed on microarrays. And this is now, I think, a well-used tool, and I know there are many centers in Mexico, some of whom we are collaborating with also, that use this technology to be able to look at differences in gene expression between two different, either disease and non-disease, or two different disease states. So it's now becoming, there are lots of publications, lots of data sets out, so that I won't need to go into the details of how these experiments are run. But essentially, there are many statistical procedures that are available for testing differential gene expression between different groups. So for the purpose of today's talk and the analysis, I will be showing you data between acute rejection and non-acute rejection, and I'll take you through different categories. So this is, as I said, one of the most expensive publications that came out of our lab. This is data that was actually developed from microarray discovery from our lab, verified within the Stanford population, and then the model was locked and tested in a randomized multi-center trial in the US, funded by the NIH across 12 different US transplant programs in pediatric and young adult patients. So the important take-home message is, you can see there is a very familiar names here of principal investigators that actually are KOLs at various centers in the US. They all contributed patients as well as blood samples that actually went into the validation of this gene panel. And this was also accompanied by an editorial, and then also discussed in Nature Nephrology that perhaps we're on the way, the future is here, that we can start to use these tests. So I'm going to now take you through perhaps what the performance of this test is. So this is the process of discovery. We used a multi-parameter approach. Again, as I said, every blood sample was paired with a biopsy sample. Every biopsy sample was read by the same centralized pathologist. That's Dr. Kambam at Stanford. So we actually had samples that were put on 3 different platforms: the Affymetrix oligonucleotide arrays, the Agilent oligonucleotide arrays, and the LymphoChip cDNA arrays. Why did we use 3 different array platforms? One of it was just the evolution of the use of different array technologies over time. As I said, this is a study that actually took 12 years. The second reason is we wanted to look at cross-hybridization across different platforms, because sometimes the data is not replicatable when you actually run samples across different platforms for every gene that is sampled. We also actually flow purified cell subsets from patients with acute rejection, as well as absence of acute rejection, and also those were processed on microarrays. Various statistical and selection criteria were put through, including cell-specific enrichment. We also, in the process of this, discovered that the white count and differential count of the patient was a very significant confounder. So a new program was actually written in Statistical Analysis of Microarrays, or SAM, a program we use for analysis of microarray data sets, which was published in Nature Methods, and that program was developed by Rob Tibshirani. In the interest of time, I can't go into the details, but perhaps I can address what it showed us in the questions. So we selected a set of about 43 genes, which essentially was the discovery set. These sets were then reduced to PCR that was tested within the Stanford cohort, and a final selection of biomarkers was produced. So about 10 genes actually performed very, very strongly by PCR. These genes were then put into a logistic regression model, and even a set of 5 could perform very well to segregate acute rejection from non-acute rejection in the blood samples. This locked model was then delivered to the NIH biostatisticians. The PCRs were all run blindly and submitted back to the NIH statisticians, who then unblinded the data at the end of the study and reported the performance specs. So that's in the 12-center clinical trial. For people in this room who are familiar with doing a lot of PCR work, you will know that there are many different platforms. And you will see that there are names in yellow. So these PCRs were actually run on the ABI 7500, the ABI 7900, the VIA 7, as well as the Fluidigm machines, which one of these machines which we have in our lab. The importance is we wanted the genes to also be platform agnostic. So it should work regardless of whichever platform you're going to test it on. But the important take-home message is most clinical labs, if you're going to make this test available, will be able to run the PCR on the ABI machine, which is actually an approved machine, and it is present in CLIA certified labs that can run assays. So this really is the hunt for a needle in a haystack. You can see that the genes shown here are not genes that have been previously studied in the literature to be tracking with acute rejection. Perforin, granzyme, FAS, granulysin, these are all cytotoxic T lymphocyte genes that we have heard about for a long time. So the big question is, did they come up in this analysis? The answer is yes, they did come up in a subset of patients. They were very high in some of the acute rejections, but were they invariant? So when you used just those genes to actually segregate out samples and you looked at their performance specs, they did not perform as well. And the reason is because of the confounder of other variants within the patients, such as the strongest confounder, there was infection and immunosuppression, as well as the white count differential that actually drove the segregation of some of these CTL genes. So good mechanistic genes, but perhaps not the best biomarkers. If you look at this, the final logistic regression model that picked these genes, picked 3 genes that were upregulated in acute rejection, DUSP, PBF1, PSEN1, and you can see 2 genes that were downregulated in acute rejection, and a combined model was built with a threshold output for prediction of acute rejection. The NIH statisticians ran these models against 23 different clinical confounders that were collected during the clinical trial. So very, very stringent analysis done by multivariate analysis to control for any other clinical confounders. They also captured 5 different biopsy confounders. So very important, this gene model picked up acute rejection irrespective of whether the acute rejection was cellular or humoral, irrespective of the grade, Banff grade of rejection. And I think as a first pass, we were very interested in finding the bigger picture of diagnosis of acute rejection. Currently, there is work underway in our lab to now use these gene panels to segregate between cellular and humoral rejection, but that was not the focus of the first study. So how did these genes perform and produce an output in the samples in the study? So this is actually data that's in the paper that is published, and I'll show you the data that comes here below, which shows the red dots are samples that are taken, blood samples taken with matched biopsies with Banff graded acute rejection. So let's just focus on the top. So these are all Banff graded acute rejection. In green are blood samples taken with biopsies that are completely stable, so no pathology, no significant pathology. Let's say all biopsies may have a 5% IFTA or whatever, but otherwise no significant pathology. So you can see that there are 3 outliers here. So there are 3 blood dots, the red dots that sit at the bottom, and these are true outliers. They were all mild Banff grade rejections, definitely confirmed as rejection. On follow-up, these are probably mild rejections. All patients seem to recover function after treatment, but they're treated as true outliers and calculated in this very impressive ROC. The important thing is that most of these green dots have very low scores. The logistic regression score from the gene set is on the y-axis from 0 to 100. The hashed line of 50, if the score is above 50, the model calls the sample acute rejection. If the score is below 50, the model calls the sample not acute rejection. You can see the majority of the stable samples have very low scores, around 20%, 0 to 20%, with some scatter. The majority of the red samples have very high scores, somewhere between 80 to 100%, but with some scatter. Note the second outlier. There are these samples here, the green dots, the samples that have very high prediction scores for acute rejection. When we went back and asked the NIH statisticians, these were all biopsies that were histologically clean. When they followed up these biopsies, these blood samples or these patients over time, they found that each and every patient within a period of 6 months developed histologically confirmed acute rejection. So this was the first time we had a clue that the blood test was not just detecting rejection when it was clinically evident by histological examination in the biopsy, but could perhaps predate the histological evolution of acute rejection. So this was actually a very interesting finding. This has again been factored in as a true outlier, but you can see despite that, the AUC is very impressive. The NIH at that point actually asked us to go back, apply for additional ERA funds, and said, we want you to now test every sample that's in your freezer. We want you to pull out the chronic allograft nephropathy, the drug toxicity, the BK nephritis, the recurrence of disease, and we want to see how this test performs in everybody else, so a true life setting in your transplant clinic, because this is false. Not everybody in your clinic is not just acute rejection or stable. So this is everybody else. So you can see here is the, I don't know whether actually the code is not very visible here, so I'll take you through it. The light blue are the CAN samples. The slightly darker blue here are drug toxicity. These darker blue here are others, and out of which this sample and this sample are BK nephritis. The others are others like cause of recurrence, disease, etc. The pink dots are actually the most interesting. So these are samples that had borderline acute rejection. So the NIH did not want us to use any borderline acute rejections in this group because the analysis initially had to be very clean. So if you see, the majority of the outliers here are actually the pink dots. So again, it fits in with the same learning that borderline acute rejection, where your serum creatinine is stable and you just picked it up perhaps on the protocol biopsy, does not meet Banff criteria. A vast number of them are actually positive for the gene signature, showing that you're picking up very early acute rejections, borderline acute rejections, and perhaps you're able to also predate the histological evolution of acute rejection. So using all of these as outliers, you can see, again, very impressive AUCs of 93% here, 95% here. So now I'm going to show you data that has not been published in this paper, which is actually going to come out in another paper, is basically what do you do when you look at samples from the same patient over time? What do you do when you look at longitudinal samples? Because the data I showed you was cross-sectional. So when you look at longitudinal samples, you look at people who have samples that have been collected over time in the first 3 months, 3 to 6 months, 6 to 12 months, and greater than 12 months post-transplantation. You can see if people are stable creatinines, they have had protocol biopsies, the biopsies are clean, the kidney is doing well, you can see that these scores are actually very low. The scatter is not a lot. Again, you can see they're around that 20% mark, so very little scatter. What happens if you actually look at a patient who had an acute rejection episode and if you happen to have samples before and after the episode? So we went back to the trial, and this is the average score. So of those dots that I was showing you in the previous slide, this is the average score with very small scatter bars, you can see, of the score in patients at the time of acute rejection, time 0. So we had 40 unique patients over here. If you went back and looked at the blood sample from those patients 3 months before, you can see the scores are almost equivalent. And if you go back to 6 months before, you can see the scores are still quite elevated, significantly higher than the scores you're seeing in patients who've never had acute rejection episode. So, and importantly, if you treat these patients, and in the trial, of course, we did not control intent to treat, so physicians gave either steroids or thymoglobulin, few patients got rituximab. So you can see there is a fall in the score with treatment, but essentially the score does not go down in everybody. So if you actually break this up, there are some people who go all the way down. But there are actually about 50% of the people, at least in this trial, where the scores remained elevated. So I think these could be very important insights into being able to use this test serially to predict the onset perhaps of acute rejection before it's histologically established, but also use it as a means to follow treatment of acute rejection. And perhaps in the patients whose scores fell completely, you don't need a follow-up biopsy, because in our practice we do a biopsy 4 to 6 weeks later in everybody, and in some it's clear and some it's not. But in the people where the scores don't fall, perhaps you should be doing an earlier biopsy, or you should be looking at your immunosuppression and perhaps giving them something different. So perhaps you are able to tell between treatment resistance and treatment sensitivity. And actually the same test, and again this is unpublished data, but this paper is also in submission, but we have 126 blood samples, again matched with protocol biopsies from heart transplant recipients. These are patients that came from a sub-study of the CARGO study that led to the discovery of the AlloMap test. And Dr. Valentine is actually the cardiologist who provided all these samples. And you can see again, this is the score here from 0 to 100%. That's the threshold for the model. The coefficients are slightly different, so the threshold is slightly different. And you can see if patients have CMV, there is really not any noise. If you can see in stable biopsies, there is some outliers, but the majority are low. But if you can see in the people who actually have biopsy confirmed rejection, and a bunch of these over here are actually 1Bs, the 1Bs also actually have very high scores. So it tells you that this blood test that you're measuring these genes on is probably reflecting a trafficking cell that is probably responsible for the kind of injury, the tubulitis kind of injury, perhaps that's happening in allograft rejection in the kidney transplant, and perhaps the commonality of this kind of peripheral trafficking mechanism means that perhaps you can start to monitor for acute rejection in different solid organs. So we have early discovery work that's happening in our lab where we have a smaller subset of intestine and a smaller subset of lung transplant patients with and without acute rejection, and it looks like the panel discriminates in these smaller numbers also for intestine and lung. So we're calling this a solid organ rejection test, or the SORT assay, because wouldn't that be wonderful if we had a blood test and we could take it as a commonality to test acute rejection in any of our solid organ transplant patients? So I think it's an interesting paradigm, and further work to segregate between cellular and humoral is underway. So let's move to the second problem, the problem of trying to understand safety. And understanding the mechanism of graft acceptance is really most important to us, because without that we are facing the problem of lifelong immunosuppression. We have lots of drug toxicities to the patient, but we also have problems with infection, malignancy, lots of meds that also are used to treat the side effects of the drugs that in fact these patients are on, non-adherence, and then of course not just the drug toxicity to the patient, but also the drug toxicity to the graft with its impact on survival. And if you actually look at multivariate relative risks for death-censored graft loss, despite the fall in acute rejection rates, there is really not much change, and the jury is out whether this is due to chronic subliminal immune activation that we are unable to pick up by just the serum creatinine, or whether indeed this is also accrual of toxicity from the drugs. So to try and understand this better, we looked at a subgroup of patients who actually met an operational definition of transplant tolerance. And in fact, I know there's, with Dr. Albaru and her colleagues, we're actually doing a similar collaboration analysis of the same phenotype of patients in Mexico. So the operational definition is absence of a destructive alloimmune response in the absence of exogenous maintenance immunosuppression in an immunocompetent host. In all of our patient populations, I think we all have 1 or 2 patients who don't take their medications and are lucky enough not to reject and are roaming around and have a kidney accepted. None of us should be promoting that these patients stay off meds, but it's the natural course that tells us that there are patients who need less meds, and those are the patients that meet this operational definition. And this is an earlier paper in PNAS in collaboration with Jean-Paul Solilou, saying that indeed if you look at peripheral blood, you can pick up genes in peripheral blood that can track with this phenomena. And actually, if you look at who these patients are in our cohort, they're really all HLA mismatched patients. In fact, in our group, we have about 30 plus patients who meet this operational tolerant category now collected from all over the world, and their mean HLA mismatch is about 2.6. These are usually all over 5 years post-transplant. In a small percentage, they've had prior history of PTLD that has caused discontinuation of immunosuppression, but the majority have just over time stopped immunosuppression themselves due to non-adherence. In our earlier subgroup, we had shown that these patients are immunologically competent because they have normal responses to flu vaccination. Interestingly, a subgroup of these patients will have a prior history of acute rejection. So again, an important paradigm shift. Just because a patient has an early acute rejection episode doesn't mean that that patient is forever at risk and is going to always reject, because the immune response can skew over time, and thus the need to develop these tests that can allow for real-life monitoring. So this is really a dendrogram from the original paper showing again those scores. This is actually a score with an output of prediction analysis of microarrays, which basically, if you just look at the green bars, so the height of the green bars basically tells you which patients show maximum scores for tolerance. So you can see the green bars are really low to absent in the other categories, but you can see it's very high in this category, showing that you can definitely find some gene panel here that tracks with this phenomena. Now, understanding the mechanism of it is, of course, a tall order. So we have, in this earlier study, to try and control for the absence of immunosuppression, we had used patients who had chronic allograft nephropathy with failed transplants, some of them were even on dialysis, so controlling for the absence of immunosuppression. We have gone on and actually controlled and used more real-life CAN patients, those who are actually on maintenance immunosuppression also, but low dose. And you can see we've actually done additional work on adjuvant arrays, and again can find very strong markers in the periphery that can actually track and separate these kind of groups of tolerant patients from CAN, as well as healthy controls. So healthy controls are included because, of course, there are also people who are not on immunosuppression. And using these kind of panels, we've actually surveyed about 212 blood samples from patients who are on stable immunosuppression. So they're on 2 or 3 immunosuppressive drugs doing well. And the question is, can you see the same kind of signature that you're seeing in operationally tolerant patients? If we go back to the slide, you can see that these green bars, in a subset of patients who were just on one drug called the MIS group, you could see that about 50% of those patients, but not all, but 50% of those patients did have high scores for tolerance. And in people who were on 3 drugs in this stable group, one patient surveyed here had very high scores, which suggested that perhaps people even on regular immunosuppression could demonstrate this kind of signature, and that maybe the signature could be used to wean immunosuppression. So we went and asked what is the incidence of that signature if we were to survey a much larger pool of patients. And we found that in fact 25% of the patients surveyed actually had unexpectedly high prediction scores for tolerance. But we found that this did track with people being on lower meds. So the people on lower meds had a higher incidence of having this tolerance signature. We do not think that's related to the lower level of immunosuppression, because half those patients did not have the score, but we think that these are people who are self-selected into moving into that more preferential category, and thus you're probably seeing that higher incidence. This is actually just a slide from the data that actually is in collaboration with Dr. Albaru and her colleagues. I think Dr. Albaru hasn't seen this slide herself yet. But basically, I think some patients in Mexico have been run on the Illumina bead array in collaboration. And you can see that we are being able to find similar kind of genes that are coming out even in their analysis on a smaller group of tolerant patients. And those genes are giving us an understanding of what the mechanisms may be, because they seem to be expressed in naive B cells as well as in some categories of dendritic cells. So inducing transplant tolerance, if the solution is that there is some mechanism that we can harness, if the solution is that maybe we can use this to titrate immunosuppression, can we test this in an induced tolerance protocol? So if the solution has to be predictable and durable, can we basically test these signatures in people who are undergoing a tolerance induction protocol? And to do this, we actually looked at a protocol for tolerance induction in kidney transplantation that is underway at Stanford by Dr. Sam Strober, Negrin, Lowski, and Dr. Scandling. And I think some of the preliminary data from this trial has been published in the New England Journal in 2 separate, I think, publications. But essentially, these patients receive a kidney transplant. These are all adults with TLI and ATG in the following regimen, as well as a donor CD34 cell infusion. And these patients are on prednisone for 10 days only. CellCept is discontinued at day 30, and a cyclosporin taper begins at 3 months. The idea was to withdraw immunosuppression around 6 months. And Dr. Strober came to us and said, if you have a gene panel that perhaps tracks with the detection of operational tolerance, the first question is that gene panel the same, and could it pick up tolerance in, again, kidney transplant patients, but in a manipulation induction protocol? And secondly, can we use this panel to therefore titrate and figure out which patients can withdraw off cyclosporin more safely? And so the monitoring for induced tolerance by their current parameters was not working as well, because their hypothesis initially was that all these patients are going to be persistently chimeric. But that did not work out, and these patients had variable levels of chimerism. So we basically did peripheral blood gene arrays on these patients. And so far, 12 patients have been enrolled. 11 patients have greater than 1 year follow-up. 3 patients lost chimerism very early, had an acute rejection episode. So these were labeled by Dr. Strober as the failed patients. And then we had 8 patients who've had persistent chimerism for more than 6 months. And in fact, all 8 to date are off all immunosuppression for a year with stable graft function. So when we ran the arrays on these patients, I think it's very important to see when we received de-identified blood samples from Dr. Strober. And you can see that these were actually samples when they were unblinded that came from the patients prior to transplantation. So again, this is the score on the y-axis. This is the prediction cutoff. And you can see that they are all below the 50%, which means they are not fitting the scores for tolerance. And you can see these were all the pre-samples. So you don't have that signature prior to transplantation, which is good. These were actually the 3 patients that had the acute rejection episode and had to go back on immunosuppression. All of these blood samples were sent at the time of the acute rejection, or these are when the patients were 1 year off drug. And so you can see these are the prediction scores from the other patients who are actually now with stable clinical tolerance. They meet all definitions and criteria and off all immunosuppression for a year. So I think the interesting first take-home message was the blood gene sets for operational tolerance and induced tolerance seem to have some overlap. So even though the mechanism of induction of tolerance was quite different, perhaps what we are monitoring downstream and actually can still serve as a biomarker. And these gene sets can at least predict stable tolerance. So I'm not going to show you the rest of the serial data from these patients in the interest of time, but summary is that gene arrays on peripheral blood samples therefore can pick up a peripheral signature for dominant tolerance. There are overlaps in these gene sets for operational and induced tolerance. And stable tolerance in this protocol cannot be predicted by chimerism alone, which means we need other tests. And the incidence of stable patients with operational tolerance actually can be quite significant in our populations and in our clinics. So to be able to develop these assays and bring them to monitor at least which patients can reduce their immunosuppression or maybe start more aggressive surveillance for viral infections, I think would be an important take-home message. And so serial measurement of expression of these genes may allow for deliberate immunosuppression minimization after transplantation. So I'd like to just take you quickly through what these mechanisms may mean, but in the interest of time, I think there's an unspoken assumption behind the tolerance industry that it is possible to manipulate the mature immune system to permanently suspend a specific response to one set of antigens without compromising immunocompetence with acceptable risk and cost. So you have to remember this is a large assumption that we are making, that indeed we can get to this point. And if we revisit the old concepts, what are really the mechanisms underlying this? I think it's still unclear. All of these mechanisms have been proposed. But if we actually look at the gene array data, what have we learned? We've learned that operational tolerance is actually an actively regulated process. So if you look at the gene expression in these patients, it's not similar to normal people. So this is global variance or change in the expression of genes over the entire chip. And if you look at normal people, there is very little change. If you look at tolerant people, there is a massive shift in a number of genes to maintain that state. And this is intermediate in variance between patients with bacterial infection and patients with lymphoma. So patients with lymphoma obviously have a large variance in their genes. So it's an actively regulated process. The tolerance signature also, it depends on what denominator you compare this to. We've seen the papers in JCI from the European Consortium and the ITN that when they actually compare tolerant patients to stable patients, patients on triple immunosuppression, there is a very strong B cell signature. If you compare tolerant patients to patients with chronic allograft nephropathy and control for healthy controls, you actually find there is a stronger T cell signature. And in fact, these T cell specific genes are enriched with reduced T cell activation genes, reduced co-stimulatory genes, and it's possible that the whole process of tolerance actually follows some earlier immune accommodation, a similar, I guess, proposal that was proposed by Dr. Starzl, that there has to be recognition of antigen for adaptation of the immune response. And maybe that explains the kind of strong also driver of some T cell signature that lies within these patients. But despite that, if you look at acute rejection and its T cell signature and what comes out in tolerance, it's not just what's present in acute rejection is absent in tolerance. There's very, very little. And it could be, I'm going to skip through this slide in the interest of time, but if you look at the genes, you can see that in fact the genes that come out for tolerance show reduced expression of Th1 and Th2 expression, co-stimulation, and cytokines. And you can see here these few genes, early genes, are actually highly expressed in Tregs. So take-home message is in all of this gene expression data, it depends on who your denominator is. You can see GITR, neuropilin, some TGF-beta regulated genes are all increased. You can see that here some NK genes can be increased, but it depends again on the denominator. So very difficult to dissect complete mechanisms from biomarkers. We also found that there's a very strong regulation for TGF-beta, and in fact 27% of the genes in this tolerance set are regulated through TGF-beta. And when Dr. Jean-Paul Solilou's group examined their patients independently, they found that TGF-beta1 serum levels were much higher in operationally tolerant patients than those with chronic rejection. And if you ask what is TGF-beta perhaps doing, you know that TGF-beta may be skewing the whole induction as well as the maintenance phase of tolerance. It's been seen in animal models. And TGF-beta, of course, also supports different populations of Tregs, as shown here. It's also possible that TGF-beta may be altering the threshold for T cell activation, because anecdotally, in the initial study that we have, and there is one patient that was 8 years off all immunosuppression, landed up having a tattoo that got infected. And the patient had a few weeks later a fulminant acute rejection episode and lost his graft after being off all immunosuppression for 8 years. So a shift in the immune response and its kind of balance, or the homeostatic proliferation of these cells, or perhaps the TGF-beta altering the threshold for T cell activation, means tolerance is really a metastable state. So if you look at, people have looked at peripheral expansion. So with the microarray data, can we get insights, and are there expanded populations of T cells? And you can look at these different cells and you can see, first of all, take-home messages, there's a lot of scatter. In the cells that look slightly different, CD25, CD4, here you can see this population of CD45RA, CD62L negative, CD4 positives. You can see there is a shift, but again, there's no difference between tolerant and healthy controls. So I think we need to look deeper and find something else that can clearly distinguish what's happening in operational tolerance, because I showed you the gene expression data. There is clearly something very dramatically different in operationally tolerant patients from just healthy controls. So though there is some expansion in tolerant compared to chronic, it's not different from normal controls. So the unanswered questions remain in tolerance. What are the exact cellular processes driving tolerance? Is it Tregs? We know it is a metastable state, and I told you about the loss of tolerance here. We really don't know what perpetuates it, and I think more work is required so that we best know also how to wean immunosuppression in patients that show this signature. We don't know when it develops, and we are in the process of serially surveying patients because most of our tolerant patients are more than 5 years out from transplant, and we don't really know what to do to promote it. We know that the tolerance signature across organs, and we have data published in liver and in kidney, the genes that come out in peripheral blood are quite different. The liver genes are very NK driven. The kidney tolerance genes are actually some B cell, dendritic cell, and T cell driven. So the last slide, I hope that maybe there's excitement that there is a future for evolved monitoring for transplant care. We need to bring in tests that test the yin and the yang. The gene arrays have helped us discover very sensitive and specific novel gene sets for peripheral non-invasive monitoring for both rejection and tolerance. These have been all reduced to PCR, so they're cheap, 24-hour turnaround. They can be situated in a local clinical lab, and actually the rejection test is actually preparing to go forward for FDA review. So I'd like to invite transplant programs here in Mexico to actually join us to help in prospective screening of your patients for prediction of these immunological signatures of activation and quiescence. And if there's interest, there's my email address, and that's a very easy lab website to remember, sarwalab.org. So I'd end by really thanking all the people in my group, our collaborators, Dr. Albaru and her colleagues in Mexico, and funding from 4 different institutes at the NIH. Thank you very much.